Hi everyone, welcome to this week's module. This week we're going to be taking a look at those two basic fundamental levels of organization in the human body. Today we're going to start off our lecture looking at the chemical level of organization, and this is where we look at those smallest little building blocks, those pieces, the Lego pieces essentially, right? The atoms that are put together to form larger molecules and ultimately the organic macromolecules that make up all of our cells. Okay, so those organic macromolecules we're going to talk about today: the carbohydrates, lipids, proteins, and nucleic acids. Then we're going to move in the second half of lecture to looking at the cellular level, okay, where we're going to look at the structure of the cells, and then next week we're going to be focusing in on how all of those cells are specialized to form the tissues, and that will lead us into then the rest of the discussion throughout the remaining weeks in this semester. So those smallest little Lego building blocks that we keep talking about, those are the atoms. And if you think back to chemistry, whatever your experience may be in high school or beyond, think back to the periodic table of elements, okay? So that's where you're gonna find the names of all of those different atoms that exist within our bodies. Okay, so those are the smallest units of matter, and they're used to build up the more complicated molecules, okay? So here we're looking at an example of a carbon atom, and you can see that there are 4 electrons here in its outer orbital, and those electrons are the negatively charged particles. Okay, and here with this example of a molecule, one that is most prevalent throughout our bodies, this is 2 hydrogens bound to an oxygen, and that is H2O, which is water. Now, the way that those different atoms are held together, so for example here, looking at the oxygen with the 2 hydrogens, is through chemical bonding. So the atoms with those energy levels that are unbalanced are going to react with other atoms that have also unfulfilled outer energy levels. Okay, and then when that happens, either atoms are going to donate and receive with one another, or they are going to share. And we're going to see the 2 different examples here moving forward. So the one example, or first example we're going to take a look at, are the nonmetal and metals. And when they react with one another, you have what are known as ionic bonds. So those ionic bonds are formed when you're going to be having a nonmetal and metal reacting with one another, and the outer energy level, you can see here with the sodium, you're going to have a donation of an electron to one, or the other atom, the chloride in this case, is going to be receiving. Okay, so you have donating and receiving of the electron in the outer orbital. One of those is going to be a positive ion, which is known as a cation, and the other is going to be known as a anion, which is a negative ion. Okay, and the way that that works is the one that's losing the electron, it's going to be known as, or it's going to become the positive ion, and the one that's receiving is going to become the negative ion. So in this case here, we can see the sodium ion is going to be losing, so it's going to become positive, right? If you're losing negativity, you become more positive. That's a good way to think about it, okay? So it's losing negativity, it's becoming more positive, and so it is now a cation. And the chloride is going to be the one receiving, and so it is going to become more negative because it's receiving that negative electron, and it's going to be now an anion. Okay, so a few key points here. So ionic bonds are formed between a metal and a non-metal. One of those is going to become a positive ion, which is known as a cation, and the other is going to become a negative ion, which is known as an anion. Now here's an example here. We were just looking at sodium and chlorine, and when they react together, they form bonds, they become sodium chloride. Now sodium chloride, that is your salt, your table salt. And one thing that's unique about salt is that if you put it into a solution like water, into a solvent, it can then easily and readily dissolve. Okay, and the reason why is because with ionic bonds, these electrons are just being donated from one to the other. And so once in solution, the water molecules that are surrounding them help to break those bonds and allow for both of these individual ions to be able to be suspended and dissolved in solution. So 3 key components at this point: ionic bonds between metals and nonmetals, you have donating and receiving of the electrons, and they can easily be dissolved in water. Okay, now when these ionic bonds are broken and these ionic compounds are dissolved in solution, those ions are then known as electrolytes. Okay, and I'm sure you've heard of this term before. Electrolytes, something that you find within performance drinks such as Powerade, Gatorade, okay? All of those have dissolved salts within them. And when you dissolve the salts inside those solutions, you're now allowing the body to be able to regenerate the different salts and the different metals that are essentially being lost through sweating, okay? So for any of you who have ever run a marathon, you know, they always have those rest stations, drink stations. Well, at those rest and drink stations, they always have something there that's going to contain dissolved electrolytes in it, right? They would never just replenish with straight water. And that's not a good idea to do because you are not actually replenishing all of those lost electrolytes that have been lost through sweat, okay? So you can see here, just looking at this nutrition label of that Powerade, we have here magnesium chloride. So if you look at the periodic table, magnesium's on the left and chloride would be on the right. So you'd have metal and a non-metal. So you know it's an ionic compound. And then calcium chloride. Okay, so you can have calcium chloride, magnesium chloride, sodium chloride is your typical table salt. Okay, so all of these different cations are going to be lost from your sweat, and then you're going to be able to replenish them by taking in the dissolved electrolytes in solution. Okay, we're going to talk about this in more detail once we get to water and electrolyte balance next semester. So I'll leave you at that point. Okay, so ionic bonds between metals and nonmetals, easily dissolved in solution. The electrons are donated from one and received by the other. And when dissolved in solution, they form electrolytes. Okay, so 4 main takeaways there. Now, covalent bonding is the other type of bonding that can occur, and this occurs between 2 non-metals. And you see this existing within our organic compounds, okay? So when we're talking about these organic macromolecules later on today, those are all organic compounds where covalent bonding exists between the individual atoms within that molecule, okay? Now, this is important because covalent bonds are much stronger and they do not dissolve in solution. And looking here at the bottom of this, this slide, you can see here we have a carbon bound to 2 oxygen atoms, okay? And if you look at where those exist on the periodic table, both within the non-metal category, all right? So covalent bonding between 2 non-metals, electrons are shared and cannot be dissolved in solution. Now, the third type of bonding we're going to talk about today are hydrogen bonds. Now, hydrogen bonds are relatively weaker in comparison between covalent and ionic bonds. Hydrogen bonds exist between polar molecules where you have a region of the molecule that is slightly charged negative and the other that is slightly positive. And what happens with water, water is unique in that you have different water molecules are going to be slightly attracted to one another through hydrogen bonding. So you can see here, just as one example, you have the slightly positive hydrogen of one and then the slightly negative oxygen of another. There is going to be a slight attraction, right? Positives and opposites attract, okay? So the slightly positive hydrogen and the slightly negative oxygen are going to be attracted to one another. And this is what creates surface tension. Okay, so if you've ever seen, you know, been at a pond or a lake and you've seen the little water bugs or water spiders that are just running across the surface there of the water, the reason that they are able to do that is because of the hydrogen bonds that exist between adjacent water molecules. Now, this also comes in and plays a role when you have water within our bodies and the way that proteins are going to interact or different parts of a protein are going to interact with the water molecules around it. Certain parts of a protein are hydrophobic, which means essentially they're afraid of water. Okay, those parts repel water. And other parts are hydrophilic. So you can have these different regions within a protein that are going to cause it to, certain parts are going to want to fold towards the interior of the protein and others are going to stay toward the outside of the protein and be exposed to water. So water, being an inorganic compound, is essential to life, most abundant in the body, and it acts as a solvent. Now, I've used this term solvent previously, but here just to define it, a solvent is a substance in which a solute is dissolved. Okay, so the solutes that we've talked about so far, were those salts, the ionic compounds, right? When you take sodium chloride, magnesium chloride, calcium chloride, and you place them, those salts, into water, which is the solvent, they are going to dissolve and dissociate. And when they do, they become their individual ions, which we refer to as electrolytes. Okay. So now we're going to take a step. We've looked at atoms, we've looked at molecules and the bonds that hold them together. And now we're going to look at how we go about building up our major macromolecules in the body. Okay, and I mentioned last week briefly, but there are 4 major organic macromolecules that are sort of the essential building blocks of every structure in our body. Okay, and these 3 of them you can find on the nutrition label. All right, and those are your proteins, carbohydrates, and your lipids. Now, the reason that nucleic acids, which is the category that covers DNA and RNA, the reason that nucleic acids are not included on the nutrition label is because they do not offer us any caloric value, okay, which means we don't obtain any usable energy from breaking them down. But what we can do is take the DNA from, say, a plant cell, break it down into the individual monomers, which are known as nucleotides, and then we can use those nucleotides when we're making new cells. When, say, you're making your new skin cells, you can use those nucleotides to help to make the DNA that's going to be residing within each of those individual cells. Okay, so we use all the different, we take all of these different parts in, break them down, and then it's the proteins, carbs, and lipids that can provide us with caloric value, so provide us with a means by which we can create energy. But the nucleic acids, they can be used as building blocks within our body, but they don't provide us that caloric value. Now, the macromolecules are the proteins, lipids, carbs, and nucleic acids. Each of those macromolecules are made up of individual parts. Okay, and if you heard previously, I mentioned that DNA, when broken down, is made up of nucleotides. Okay, that's an example of the monomers that are acting as the building blocks that build up your larger polymer, your DNA molecule. Okay, in this example here, what we're looking at is individual monomers that are making up a larger polymer. Now, the polymers, okay, poly just means many, right? So more complex molecule. The polymer is a generalized term for your macromolecules. Okay, and the monomers are the building blocks that build up those macromolecules. So here we're looking at an example of a polymer that's known as a protein. Okay, this is in its simple structure where you have 3 monomers, and in this case, amino acids. 3 amino acids that are linked together to create that complex polypeptide chain. So in each case, whether you're looking at proteins, fats, carbohydrates, or nucleic acids, each of those macromolecules is made up from smaller monomers, individual subunits. Okay, and we're going to go through each of those as we progress through the rest of the lecture. Now, we can store up those monomers and form larger polymers to be stored in the body, or we can take the larger polymers and then break them down into the smaller monomers to be able to use those individual parts throughout. So what we're going to look at here is just an example of how we would build up a larger carbohydrate and then how we would break down the larger carbohydrate to produce our individual monomers, which in this case are your glucose molecules. So here we're looking on the left at the process of building up, okay, a more complex macromolecule. And here on the right, we're looking at the process by which we break down the more complex macromolecule into individual monomers. Okay, so here, this process of building up is known as dehydration synthesis, and the process of breaking down is known as hydrolysis. Okay, so the name comes from what's happening with water and how water interacts with this reaction. So first off, this is the example we're going to start with. And I want you to sort of forget that we're coming from this point and linking to this point, okay? So this is our starting point. We have just taken in large amounts of glucose. So somebody has just drinking a very sugary drink. Say we've just drinking a Coke, okay? And we've taken in a whole bunch of monosaccharide there, okay? Those individual units, we're going to use them to make ATP, but if we have excess glucose that's not going to be utilized, we can then take that excess glucose and we can combine it with other glucose molecules and store it up as a more complex carbohydrate known as glycogen. Okay, that's one example of a polysaccharide that we store up within our livers and our muscles to be able to store up that excess glucose that we've taken in dietary, and then store it up to be used for energy later. Okay, so this here first, we've looked at a dehydration synthesis reaction. We've taken glucose molecules and we're linking them up to build a larger carbohydrate, the glycogen molecule. Okay, and during this process, what we're doing to link these individual monomers up is that we are removing water, okay, from one end of one. So this is a glucose. Okay, from one end, we're removing a hydroxyl group, OH, and then from the other, we're removing a hydrogen. And when we do that, you have your H2O. Okay, so we're removing a water, which is why it's known as dehydration, and we're putting it together, synthesis. So it's kind of a dead giveaway, right? If you hear the term dehydration synthesis, you're thinking we're losing water from the molecule and we're putting it together. Okay, so now we're able to link up those glucose and we're able to store them so that later on, if we're fasting or starving, we're able to then break down that glycogen and have our individual glucose molecules available so that we can produce energy from them, okay? So this first step, we've eaten something that's high in sugar, we take all that extra glucose we don't need and we store it up for future use, okay? That's known as dehydration synthesis. Now, the process of taking that larger polymer, the glycogen molecule, and breaking it down, we actually require water again to be able to help to break those individual bonds of those individual glucose units. Okay, so the water comes into play, is going to break those bonds, and now we're left with an individual glucose that can be then used to make some energy for us. This process is known as hydrolysis because hydro referring to water and lysis referring to breaking. Water is actually breaking those bonds that we formed previously so that we can use the individual monomer. Okay, so building up macromolecules, we go through a process of dehydration synthesis, and breaking them down, we go through hydrolysis. And everything there sort of just summarized for you, what's happening with water and what you're doing. So now that we know how we build them up and how we break them down, we're going to take a look at the four main classifications of macromolecules. So the first class of macromolecules are the carbohydrates. And as I just mentioned, we were looking at how to take a complex polymer, right? Our polysaccharide was the glycogen molecule. That's just a specific example. And what we were doing through hydrolysis was breaking all of the individual bonds so that we could have each glucose released so that we could produce energy from those glucose molecules, okay? So the polysaccharide is just made up of poly, many, okay? Lots of sugars. Saccharide defined means sugar, okay? Monosaccharide, those are the basic monomer building blocks. So the monosaccharide is a single sugar. Some examples we'll go through on the next slide. Glucose, fructose, and galactose. Those are your three main monosaccharides. Now a disaccharide is two sugars linked together. And we're going to look at the two examples that we'll talk about in the next slide. And then your polysaccharide is multiple, so more than two, okay? Multiple sugars that are linked up together to create a complex carbohydrate. And so the example we just talked about was glycogen. Okay, so glycogen is essentially just linked up glucose molecules. All right, we store that in our liver and our muscles. And the other example is plant-based. You've probably heard of it, known as cellulose. That's another example of a polysaccharide. One thing to add is all carbohydrates are made up of carbon, hydrogen, and oxygen. If I didn't mention it before, the four main atoms found in all organic macromolecules are carbon, hydrogen, oxygen, And then in proteins and nucleic acids, we have nitrogen. All right, so in carbs, CHO, you have your carbon, hydrogen, and oxygen. So the monosaccharides, those are the basic unit carbohydrate molecules. Examples are glucose, fructose, and galactose. The disaccharides, the double sugars, two examples we're going to talk about right now are sucrose and lactose. So lactose, I'm sure many of you have heard, some of us are lactose intolerant. Lactose is a disaccharide. It's a sugar that's found in milk products. Okay, and so lactose, we require a specific enzyme known as lactase to break up that disaccharide into its individual monosaccharides. So remember I said a disaccharide is made up of two monosaccharides. So lactose is made up of galactose and glucose. All right, and so we require the lactase enzyme to break up that bond between the glucose and galactose. And we'll talk about lactose intolerance a little bit later. Sucrose is made up of glucose and fructose. Okay, and sucrose is pretty much your table sugar. And then lastly, the polysaccharides we talked about, glycogen, that's the one that we use to store up monosaccharide units so that we can use them later. And we store those typically, you've probably heard of liver or muscle glycogen. And then the other example was cellulose found in plant products. Now the next classification of macromolecule we're going to look at are the lipids, and the lipids include fats and oils. Okay, first off, we're going to look at one lipid structure, which is known as the triglycerides. Now it receives its name from the fact that there are three fatty acid tails that are bound to a glycerol backbone. Okay, so tri meaning three, triglycerides. Now triglycerides, this is the form that we typically, if we're ingesting fat, this is how we are going to be ingesting it. We then will break it down into individual fatty acids, and we can burn those fatty acids metabolically, so to produce energy, or we can store up those fats in the form of triglycerides. Okay, so triglycerides is the most common form that we sort of interact with from a dietary perspective of fats, where we are taking in the fatty acids and then storing them up in our bodies, in our fat tissue or adipose tissue, as triglycerides. Now the next example we're going to take a look at are the phospholipids. Okay, so phospholipids are everywhere in our body because they form the cell membrane. All right, so the phospholipids, let's just jump ahead this slide so I can show you sort of a blown up image here looking at how the phospholipids are oriented to form that cell membrane. Okay, so remember each cell, this is just your typical animal cell, but in our bodies, we have specialized cells, muscle cells, epithelial cells, blood cells, bone cells. All of those cells, each of them, although they're unique in structure and function, they all have some sort of cell membrane that's going to divide the inside of that cell from the outside of that cell. Okay, and the inside we call the intracellular environment, and the outside we call the extracellular environment. Now, something that's unique about these phospholipids, and let's go back, is that they have two different regions to the molecule. Okay, so triglycerides, remember, had three fatty acid tails. Well, the phospholipids have two fatty acid tails, but the key here is that these tails are made up of fatty acids. So fat, what happens when you pour oil into, say, a glass of water? Right? Those two are going to repel from one another, and you're going to end up looking at two different layers. And typically, the oil is going to float on top of the water. So what happens here, because we have this region of the phospholipid that is known as hydrophobic, meaning it repels water, when you have these phospholipids arranged, creating a cell membrane, those tails are going to orient themselves so that they're facing each other, which creates a hydrophobic environment that is the plasma membrane. Okay, and now if you look at the phospholipid, the head region, that's made up of a phosphate group, which is hydrophilic, okay, which means it attracts water. And so the heads end up on both the intracellular and the extracellular environment. Okay, so let's take a look at this image again. See all these blue spheres? Those are all the head regions. And you can see the the two fatty acid tails from each oriented to the internal aspect of the plasma membrane. Okay, so all of the heads are going to orient themselves to the either extracellular or intracellular side, right? Because they attract to water. And then the fatty acid tails are going to be orienting themselves to the internal part of the plasma membrane. So this creates a region here within the plasma membrane that is hydrophobic. Okay, so anything that is, any molecule that's hydrophilic won't be able to simply just pass through the cell membrane. It can't just move from outside to inside or vice versa. And so there are different channels, there are different transporters that can help to move molecules that are hydrophilic from the external environment into the intracellular environment. Okay. Now, the third example of our lipids is our cholesterol molecules. Okay, and we find these in many different places in the body. We have cholesterol within our skin, and the interaction between UV rays and the cholesterol molecules in our skin is what actually can produce vitamin D. We also have cholesterol molecules that are located within the cell membranes. And these cholesterol molecules act to provide flexibility within those cell membranes. So you can see them there, there, and so on. And then cholesterol is also used as a backbone for a group of hormones known as our steroid hormones. Okay, so estrogen, testosterone, cortisol. There's other examples that we'll go through once we get to the endocrine system. But essentially the steroid hormones are fat soluble because they are made up of a cholesterol backbone. So how that comes into play: when you have a steroid hormone that needs to get into the cell to interact with the, you can see it up here, the nucleus, which is the control center, that steroid hormone can simply pass through because the steroid hormone is going to be bound to a specific protein that's going to take it around through our blood. And then as soon as it needs to get into a cell, it can simply pass through that cell membrane because it's made up of fat, right? The steroid hormone is made up of cholesterol, and this internal region of the plasma membrane is hydrophobic. And so steroid hormones have the ability to just pass through and simply act on the nucleus to tell that cell what it needs to do. There's other types that are made up of proteins, and those non-steroidal hormones, they cannot simply, and there's a different mechanism that they can use to pass into the cell. All right, our third group of macromolecules that we're going to look at are the proteins. Okay, so proteins, as we've already looked at, are made up of amino acids. So the amino acids are the monomers. And when you link up the individual amino acids together, they form the more complex polymer, and the polymer is the protein. Now, it doesn't end here, right? We looked at the example where we had 3 monomer amino acids that were linked up together forming a protein. Now, that's just a primary structure of a protein. Based on how each of these amino acids is going to be lined up in this polypeptide chain, the next structure, known as the secondary structure, is either it's going to start folding into what's known as beta pleated sheets, or it'll start twisting in different regions, which is known as a alpha helix. Okay, so this secondary structure, either the folded sheets or the twisted helix, this is the next thing that's going to happen to give this protein its shape and ultimately define its function. Okay, so really it comes down to how there's 20 different amino acids and how they are all lined up in a row. That's going to determine how this protein is ultimately going to fold and then fold further to create its final protein structure and therefore its function. All right, so the secondary structure will start to fold into these beta pleated sheets, and certain parts will start to form the alpha helix. And then you can see here, once you've formed that secondary structure, there's a tertiary structure that's going to take place where then that polypeptide is going to then be further folded into more of a globular type of structure, like a 3-dimensional structure. Okay, and this 3-dimensional structure here is going to have certain regions that are, as I mentioned, hydrophobic, and those are going to tend to fold towards the interior of the protein, and others that are hydrophilic that will stay towards the outside of that protein because they're going to be exposed to the water that's in the extracellular and intracellular spaces. All right, now if we take multiple globular proteins and we group them together, we form what's known as a quaternary structure. So this is a more complex protein structure. We see this with the hemoglobin molecule. So hemoglobin, found in our red blood cells, responsible for transporting oxygen and carbon dioxide in our blood. Hemoglobin is made up of 4 of these individual globular proteins. Okay, so we have a primary structure of a protein that's just simply linking up. Think of it as like a strand of pearls. And then finally, that strand of pearls, based on the arrangement of how these amino acids are ordered, is going to start to then fold or start to twist. And then all those folds and twists are then going to then further fold into a globular structure, the tertiary level. And then if you have more than one globular put together, that's the quaternary structure. All right, now we have different types of proteins. Remember, they're made up of amino acids. They have primary, secondary, tertiary, and quaternary structures. Some are referred to as structural proteins, and others are known as functional proteins. An example of functional proteins are enzymes. And one example I already mentioned was the lactase enzyme. Okay, so functional protein meaning that they have a specific function that they have to perform. So first off, structural proteins, you have certain ones that offer strength throughout our bodies and stability. So collagen is one type of protein that we're going to look at next week when we're looking at tissues. Collagen is a fibrous protein holding tissues together. And then keratin is one. I'm just going to change this laser pointer because my mouse keeps going away. Keratin is a protein that is found in our nails as well as in our hair and also in our epidermal cells. Okay, and it forms a tough and waterproof layering. So our hair, nails, as well as the outer part of our epidermis. So with enzymes, enzymes are functional proteins. And the whole goal here of an enzyme is to either to help catalyze a reaction, whether it's putting your macromolecules together or whether it's going to be taking for example, a disaccharide like lactose and breaking it apart. So the example that's shown here is taking molecule A and molecule B and then assembling them together. So the enzyme is a functional protein that is helping this whole reaction of bonding these two together to take place. So with lactose intolerance, we mentioned this before, the lactase enzyme is going to be doing the opposite of what you're seeing in this diagram. So with a lactase enzyme, it's going to be taking the disaccharide, which is made up of galactose and glucose, and it's going to be catalyzing the reaction, the hydrolysis reaction, to break that bond up between the glucose and the galactose. Okay. Now what happens with lactose intolerance is that usually these individuals lack the lactase enzyme in their bodies. So you can take, like lactate, for example, is a form of the lactase enzyme that you can take before ingesting anything like a dairy product that would be high in lactose. And what it does is it provides your body with the enzyme at the time to help break it down. Now, if you have a lot of that lactose left in your small intestine, because at that point, at the beginning of the small intestine, the lactase enzyme should be breaking lactose down. Okay, if you have the lactose left over, the bacteria within the intestine start to feed off of that sugar. And then what happens is excess gas production occurs as a result of them breaking, ingesting and then breaking down the sugar themselves. Now, the last class of macromolecules we're looking at are the ones that don't provide us with any nutritional gain. However, they do provide us with the building blocks in order to produce our own DNA and RNA molecules. All right, so nucleic acids is the macromolecule that sort of a classification for both our DNA as well as our RNA molecules. Now, they're made up of the monomers nucleotides, and the nucleotides contain 3 regions. You have a phosphate region, you have a sugar, okay, and you can either have a deoxyribose or a ribosugar, and that's what dictates whether it's a DNA molecule or an RNA molecule. So that's where it gets its name from. DNA stands for deoxyribonucleic acid, whereas RNA stands for ribonucleic acid. So if you're looking at a DNA molecule, Well, we know we are looking at a nucleotide in our DNA because we have a deoxyribose sugar in this image here. Okay, and then added on and attached to the first carbon of that sugar, we have what's known as a nitrogenous base. And there are 5 different types of nitrogenous bases within our DNA. There are 4. And then substituted out in our RNA, instead of thymine, we have a uracil. Okay, so we have 4 different types of nucleotides, and that's what makes up our DNA. Within our DNA, we have adenine, guanine, cytosine, and thymine. And then with RNA, you're looking at adenine, guanine, cytosine, and uracil. So a big difference, as I mentioned before, between your DNA and RNA as well, is that the sugar, you have a hydroxyl group here with the ribose sugar in your RNA. And then the deoxyribose, you're missing that oxygen, hence why it's called deoxyribose. Okay, so different sugars. You have the nucleotides A, T, C, and G, your adenine, thymine, cytosine, and guanine. Okay, so that's your example here of your DNA with the double strands. You have the nucleotides all lined up in a sequence on one strand, and then on your complementary strand, running in the opposite direction, you have the complementary base pairs. Okay, and one of the keys is that your adenine will always bind with a thymine in your DNA molecule, and your guanine will always be matched up with a cytosine. Okay, and with the RNA, your ribonucleic acids, RNA is a temporary working copy of a specific portion of your DNA, and that is utilized to be able to make a specific protein. And so with RNA, big difference, you have a uracil nitrogenous base instead of thymine, and a ribose sugar instead of a deoxyribose sugar. Okay, in this second half of the module, we're going to be focusing now on that next level up in terms of organizational hierarchy. We're going to be looking at cells. Okay, so remember, when those macromolecules are put together in a specific way, you then create your cellular level of organization. So we're going to start off looking at your typical basic animal cell, and I'm sure most of you have already seen this or been taught this at some point in your educational history. We're going to look at the basic structures as well as functions of our cells. Next week, we'll get into looking at the specialized cells that make up the individual tissues in our body. Okay, and later in this lecture, we're going to move on to looking at then how we can move substances into a cell and out of a cell. Because remember, we have to cross that hydrophobic plasma membrane in order to bring something in or to remove something from the cell. Okay, so today we're going to look at cells just in general. Next week, we're going to move into looking at the specific cells that we have in our body. Specifically, we'll look at epithelial cells that make up our epithelial tissue. We'll be looking at muscle cells. We have skeletal muscle, smooth muscle, and cardiac muscle cells that all make up both our skeletal muscles, all the other automatic muscles in the body, as well as our heart muscle. We'll look at nerve cells that make up our nervous tissue, and then blood cells, just one example of the many cell types that make up our connective tissues. Now, this is probably familiar to most of you. You've probably seen your basic animal cell at some point, okay? All of our specialized cells, whether it's epithelial, nerve cells, muscle cells, or any of those found in connective tissue, all of those cells are specialized in terms of their structure, so they won't look like this. And they're specialized in terms of their overall function, and usually structure lends itself to its function. One of the things, though, that is going to be key here is understanding these main components of a typical animal cell. Okay, this is sort of just your generalized cell structure. Okay, but one thing that is shared by all of our specialized cells is they all have the same components. Some cells, like for example, your cardiac muscle cells, are going to have far more mitochondria in them, and that's because the mitochondria are producing ATP and making energy. And think about your heart; it's always contracting, right? It's always on, and so you have more mitochondria in there to allow for those cells to be able to always function. So that'll be the big difference. Okay, is how much of certain organelles might be available in certain cell types. Okay, but today's focus is on generalized cells and looking at their structures and looking at the functions of those structures. Okay, so the basic cell structure, every cell is going to have a plasma membrane. So we just looked at that previously where we saw the phospholipid bilayer, right? The phospholipids that orient themselves to create that outer plasma membrane. And remember, the tails, the fatty acid tails, all direct towards the interior of this membrane, right? So this layer that I'm tracing along here, this is a hydrophobic region. Hydrophobic meaning that it repels water. Because everything outside the cell, water is the main component, and inside the cell in the cytoplasm, water is also a main component. All right, so every cell is going to have a plasma membrane made up of those phospholipids. And then inside the cell contains cytoplasm. And think of cytoplasm almost like as a jelly-like medium through which everything is suspended and able to move throughout the cell. Okay, we also have what's known as a cytoskeleton. There's specific filaments that are capable of, transport as well as providing structure to the cell. And then there are organelles. Okay, and the organelles, these are known as membrane-bound organelles. Each of them is like a little machine that has a specific function inside of each of our cells. And that's why the cell is known as the basic structural and functional unit of the human body. And that's because in and of itself, this cell is operating on its own. Okay, and then when you put them all together and they're all working on their own but working collectively together, that's where you create your tissue. All right, so basic cell structure, plasma membrane that surrounds the internal or separates the internal environment from the external environment. And inside the cell, we have cytoplasm, which is like the jelly-like medium that suspends everything and allows for movement within. The cytoskeleton that's also allowing for movement, but also maintaining structural support. And then the organelles that are each doing their own job to help the cell function overall. Now, one thing that I want to mention here is that the plasma membrane is selectively permeable. Okay, so selective permeability is a phenomenon by which certain things are allowed into and out of, and certain things aren't unless they are transported or assisted in some way to be brought in or removed from the cell. Okay, so the cell is able to control what comes in and what goes out. That's what selective permeability is referring to. Okay, and here's one example of a way in which the cell is able to regulate what's moving in and what's moving out. And this is an example of a protein channel. Okay, and so protein channels are specific to specific ions, specific molecules, and we have different channels that will allow for the passage of specific substances either into or out of the cell. Some of them are voltage gated, which means they only open when a certain voltage resting membrane potential is reached. And then they open and they will allow a certain type of ion to move in or out of a cell. Okay, that's just one example. We have other transporters that will actually, there's co-transporters that will help to bring glucose molecules into the cell. The cytoplasm, think about it just as that jelly-like substance that you would find inside the cell, okay, the jelly-like medium. And then the organelles all suspended in there, the little organs by definition, but the little machines, think of them as that. Okay, so we're going to move on looking at specific cell organelles here. There is a worksheet that I would have you download and start to fill in, where you have the names of the different organelles or cell parts in one column, and then another column for the function. Okay, so that's going to be the key takeaway from this portion of the lecture, is that you're able to walk away with knowing the different cell parts by name and then knowing what each of their individual functions are within the cell. So first off, we're going to start off looking at the nucleus. Now, the nucleus is known as the control center of the cell, and that's a good sort of way to define its function. It's surrounded by a nuclear envelope, which is a double-layered membrane you can see here. And inside of the nucleus is where all of the DNA, all of our deoxyribonucleic acid is stored, right? So your DNA is your genetic code. So every single cell in your body is going to have an exact copy of your DNA, which is going to set out, what type of proteins to make, how much of those proteins to make, and so on. Now, the nucleus also, this envelope, contains these nuclear pores that you can see, and this allows for your messenger RNA molecules to be able to leave the nucleus once transcription has taken place. Within the center of the nucleus, we have a region that's known as the nucleolus, and this is where the ribosomal RNA is stored. And ribosomal RNA is essential for making ribosomes, and we're going to talk about those in a little bit, but ribosomes are key for protein formation. Now, all of the DNA that's stored up within the nucleus during the cell cycle, all of it is in the form of chromatin. Okay, so think of chromatin almost as like string-like, just like spaghetti. Think about a bowl of spaghetti. Can you picture that? Okay, so that's your DNA in its uncoiled, uncondensed form, known as chromatin. Now, when it's time for the cell to duplicate, all of that chromatin is going to be condensed as chromosomes. Okay, and those chromosomes then are going to go through the process of duplicating, and then they are going to divide, and the cell will then have the daughter cells will have equal chromosomes from the original cell. Okay, each cell has 46 chromosomes within the nucleus, with the exception of our sex cells. So the gametes each contain half. And so the spermatozoa contain 23 chromosomes, and the ovum would contain 23 chromosomes. And when the sperm fertilizes an egg, the 23 and the 23 then come together, and you have a complete 46 chromosomes in that fertilized egg. So I mentioned ribosomes previously. Remember, the ribosomal RNA leaves the nucleolus, which is inside the nucleus, and then forms the ribosomes that are necessary to be able to make our proteins. Okay, so first thing to remember, I always think about this, ribosomes, think ribs, protein. Okay, so ribosomes are the protein factories. Some of the ribosomes are free floating, and others are going to be attached to the endoplasmic reticulum. We're going to go to an image of that so you can see that here, okay? So some of the ribosomes, you can see all of the sort of orange spheres, those are all symbolizing different ribosomes. And so you can see that some of them are just suspended freely within the cytoplasm, and others are bound to the endoplasmic reticulum, which is this network, tubular network are connected directly to the nucleus. Okay, the rough endoplasmic reticulum is responsible for protein production, and then further on with the smooth endoplasmic reticulum, it's responsible for producing carbohydrate and lipid products that are key for formation of new cell membrane. A key here in the definition between the two, the rough endoplasmic reticulum is known as the rough because it has ribosomes attached to the surface of it. Okay, so when you're thinking about functions, rough endoplasmic, why is it rough? Because it has ribosomes. What do ribosomes do? Produce protein. So that's the function of the rough endoplasmic reticulum, is protein production. Whereas the smooth endoplasmic reticulum, there's the absence of ribosomes. And so it's making the other macromolecules. So it's responsible for making carbohydrates as well as lipids that can be attached onto different proteins, or that can be used to make new membrane as well. The Golgi apparatus is a flat-like structure where there are multiple sacs that are almost compressed within one another. And the whole role here is to further package and modify the proteins that have been produced previously. And so one of the ways that this occurs is that you have the incoming protein that has been produced here by the rough endoplasmic reticulum. Okay, it's then released through a vesicle which contains that protein. It then will then release that protein into the Golgi apparatus, where it's going to go through some further modification. So this is where sometimes the tertiary structure is going to take shape. So where that folding, remember we talked about if you take your secondary structure and then you start to fold it into a globular protein, that's where that tertiary structure and even quaternary structure can take place. And then also further modifications can take place because sometimes our proteins will have carbohydrate pieces that are attached to them, and they're known as glycoproteins. And then you can also have other proteins that need different fat molecules attached to them, which are then called lipoproteins. Okay, so whether glycoproteins or lipoproteins, either of those are modifications of the original protein. And so that's the job of the Golgi. So think about it as almost your packaging and modification system. All right, so the nucleus, let's just review a few of these. Nucleus is where you're storing your DNA. Then you're going to be taking a copy, the messenger RNA, which is going to leave the nucleus through the pores and then enter into the rough endoplasmic reticulum, where that working copy of your DNA is going to be read and a protein is going to be created. Now that the protein's made, it then sends the protein off, and the protein goes to the Golgi apparatus, and that's where that protein is going to take shape, is going to be paired with other proteins to make the quaternary structure, and may be modified by adding a carbohydrate or fat molecules onto it. Okay, and then the final goal is either use within the cell or can then be sent outside of the cell to be used throughout the rest of the body. All right, we mentioned the mitochondria at the very beginning, where we talked about them larger in number in your cardiac muscle cells because they are always going to be contracting, right? They never take a break. So mitochondria are the powerhouses of the cell. This is where ATP, or usable energy, is going to be produced. This is where glycolysis will take place. And then finally, pyruvate, which is that final molecule from glycolytic cycle. Pyruvate will be oxidized in the mitochondria, and it will enter into the Krebs cycle and electron transport chain, which might be familiar to you from previous study. We won't go into any of those in any detail, just understand the mitochondria is taking and producing that ATP that's necessary for all of our cells to be able to function. Hence why they are known as the power plants of the cell. Right, some other organelles that we'll just briefly talk about. Lysosomes. These are like the garbage collectors of the cell. They are their own organelle surrounded by an outer membrane. They contain digestive enzymes, and so they're able to take in either broken down cell parts and break them up further, or they also have a protective function in which they can actually digest any microbes that may have been able to enter into the cell. Right, so think of them as just the garbage collectors. All right, so now moving on, we're going to take a look at cell extensions. A couple of them: microvilli, cilia, and flagella, I believe is the last one there that we'll talk about. Now, certain cells in our body contain microvilli on the surface of them. Once we get to looking at the digestive system, this is going to become much more apparent, okay? At this point right now, it's understanding the basic extension and knowing what it's there for, okay? On the surfaces of the cells found within our small intestine, because one of the roles there in the small intestine is the absorption of nutrients. Okay, so microvilli are those small finger-like extensions of the plasma membrane helping to increase the absorptive surface of the cell. Cilia, these are hair-like extensions, and this can be used by a cell to sense its surroundings, but also, this is key, capable of moving in unison in a wave-like pattern. Cells that line the inner portion of the fallopian tube, they are always moving in a wave-like manner, which you can see here. Okay, this is an example of that ciliary function here, where it actually they beat, and they're all moving in one direction. Okay, and the whole goal here is that when the ovum is within the fallopian tube, the cilia that are lining that fallopian tube are actually beating in the one direction, moving and propelling that ovum down the length of the fallopian tube towards the uterus. Okay, so these cilia are extensions that are actually moving and helping to produce movement. We also have cilia lining our upper respiratory tract. So that's what actually helps to propel any phlegm or mucus that has caught particulate or any microorganisms, and helps to get it up and out of the body. All right, and then lastly, the flagella. Flagella is like sort of a tail extension on a cell. The only cell in the human body that's capable of locomotion is the spermatozoa, so moving from one point to another. So the flagella moves around in a whip-like fashion, and within the spermatozoa, there's a lot of mitochondria that help to power that flagella to be able to propel the spermatozoa. All right, and as I mentioned before, every human cell has a specific function. Specialized functions of different cells depend on the number and types of organelles found. So this is the example I've been talking about a few times over so far. Our heart muscle or cardiac muscle contains a large number of mitochondria compared to skeletal muscles. And this is because the cardiac muscle is always contracting, right? So we need a lot of powerhouses in those cells to produce all the energy needed to keep the heart contracting. Now we're going to finish the lecture looking at how we can actually move substances across the cell membranes. Remember those cell membranes, they are hydrophobic in their interior. Okay, so you're going to need specific processes to move certain types of substances through, whereas others are going to be able to simply pass through. Okay, so first off, we're going to look at generally how substances, water, and different molecules are transported across membranes. Okay, and there's two main types of transport processes. The first one we're going to talk about, passive transport processes. And they're known as passive because they don't require added energy. Okay, and the reason that they don't require any added energy is because they are resulting in movement down a concentration gradient. Okay, so if we're looking at an example of, say, well, here's an example of diffusion. Okay, we breathe in oxygen. That oxygen, once it gets down into the deepest parts of our lungs in the alveoli, because the concentration is so much higher in our lungs than in our blood at that point, the oxygen is simply going to diffuse. It's going to just passively move because it's always going to move from an area of higher concentration to an area of lower concentration. Okay, so let's look at some examples here. So first off with diffusion, as I just mentioned. So with diffusion is a passive process by which molecules or substances are going to move from an area of high concentration to low concentration. So in this case here, we're looking at a solution where this membrane that's been placed between the two different compartments, it's permeable to both water and to the solute that's dissolved in that water. Let's just say for this purpose, the solute here is sodium chloride because we're used to that, right? We talked about that before. So you've dissolved some salt in water. You've got a higher concentration of salt. So this is the osmotic concentration is a lot higher here, 20%, versus over on this side, it's 10% salt. So if you know that this membrane is going to allow both the salt and the water to move back and forth between compartments, what's going to happen is that you're going to have an equilibrium achieved because the water is going to be able to move into the more concentrated solution to help balance that out, but also your solute is going to be able to move to the other compartment to be able to help balance out the concentration there. Now let's compare that to the process of osmosis. We still have where you're moving down the concentration gradient, but the key here is that this is what happens with our cells. Okay, with osmosis, you're looking at diffusion across a selectively permeable membrane. So in this case here, we're looking at something, some sort of solute. In this case, it's albumin, which is a blood protein that's dissolved or that's suspended in solution. So we have a higher concentration on the right and a lower concentration on the left. The membrane separating these two compartments is permeable to water, meaning water can freely move back and forth, but the protein can't. And this is what actually occurs with our cells, with the cell membrane, because remember, the cell membrane is selectively permeable, meaning only certain substances are capable of passing through readily. Others require something to actually bring it into or remove it from the cell. So in this case here, we're looking at how water can simply pass into and out of our cells. And this is why when you're running and you're sweating a lot, you need to be replenishing with some sort of beverage with electrolytes and not simply water. So follow me here. With osmosis, we're looking at the passive movement, but now it's just the movement of water, okay, because the certain solute cannot cross the membrane. So in this case, if you have 10% albumin and 5% albumin on the left, water is going to want to move into the more concentrated solution to help to balance that out. But what's going to happen here is that you're going to have more water in this right compartment for the amount of albumin that was there, because the albumin can't move out from there because this is selectively permeable. Okay, only water can pass through. So whereas our other case of simple diffusion, where you could have water moving and the solute moving, we end up with water balance is maintained from inner and from left and right, and the solute concentration is balanced. But here, because the solute itself can't move, you have a net movement of water from the left compartment to the more concentrated compartment on the right. So this is what can happen. If you sweat out a whole bunch and you're only taking in water, your extracellular compartment becomes more diluted than your intracellular compartment. And so what can happen is that the water is going to move into the cells, causing them to swell, and in extreme cases can actually cause them to burst because too much water has moved in. Okay? So osmosis is taking a look at diffusion, but across a selectively permeable membrane, such as our cell membrane. Filtration is the movement of water and solutes that's caused by hydrostatic pressure. So hydrostatic pressure is essentially blood pressure inside the capillaries, right inside the deepest parts of your kidney. Okay, so hydrostatic pressure is the actual, think about it as water pressure inside of the blood vessel. And so the higher the pressure inside, think about it like a hose, right? The higher the water pressure inside the hose, the more water that's going to be flying out of it. Similarly here with filtration, the higher the hydrostatic pressure, the more that will filter into the nephron, which is the portion of the kidney involved in filtration. And this is always going to occur down the pressure gradient. So from an area of high pressure to an area of low pressure. All right, so those are the passive processes. So we covered diffusion, osmosis, and filtration. Now we're going to take a look at the active transport processes that help to actively move substances across the cell membranes. Okay, and the key here is active, they require the use of ATP because we are moving substances up the concentration gradient. So that means we're moving from an area of low concentration to high concentration. Okay, thinking of salmon, you know, like that have to swim upstream against the natural flow of the river, right? It's the same idea. They're having to put in lots of energy to get up that concentration gradient. Similarly here with active transport processes, we're also going to be moving against concentration gradients. So you need ATP, you need energy to actually make that happen. So the three examples, ion pumps, and then phagocytosis and pinocytosis, which are the ways that a cell is capable of taking in both eating as well as drinking. Okay, so taking in, sort of think of it as food, and then taking in water. All right, so the ion pumps first. One of the ones that we're going to focus on when we look at the nervous system is the sodium-potassium pump. And the sodium-potassium pump is responsible for creating a concentration gradient where it pumps sodium outside of the cell and it brings potassium inside the cell. And so what we end up with is a situation where we have a higher concentration of sodium outside of the cell and a higher concentration of potassium inside the cell. But the ion pumps are actually actively moving ions against a concentration gradient. Okay, so they're using ATP. ATP powers the pump, sodium's pumped out of the cell, potassium is brought into the cell. All right, and then lastly, phagocytosis and pinocytosis. Because we're drawing substances into, you can see the image here, actually helps. If a cell is going to take in through phagocytosis, it's going to be actually engulfing that, pinching off a portion of that membrane and drawing that in into the internal component of the cell. So that requires energy for the cell to be able to perform all of those functions. All right, so with phagocytosis, to eat, phage, and then Pinot Grigio or Pinot Noir, right? No. But pinocytosis is to drink, right? So any kind of dissolved substances or incorporated fluids. Okay, and that brings us to the end of this module. I look forward to seeing you all in the tutorial this week. Please, if you have any questions, let me know. Send me an email and I'll get back to you as soon as possible. Otherwise, we'll see you in tutorial. Take care.